Hello everyone. Today we're going to be talking about Are You Infected? A group project from us. A team of 4 students, Eric, Ethan, Isha, and Nuri. We're students from Dalian Memorial School, Hippo. This presentation will discuss the following shown here. Firstly, we will be discussing the problem with using normal RT tests to identify for viruses. The modern RT tests we use every day are usually overpackaged and including more plastic than the actual test itself. They're also very inaccurate and inefficient in testing for viruses, usually being able to only detect one virus at a time.
Research shows that in 2020, more than 50 tons of waste from RT tests are thrown away every day. RT tests are known to only have an average of 80% accuracy on detecting COVID. To overcome these issues, we can use an Arduino PCR machine to test for COVID-19 with much higher accuracy and less waste. The PCR machine will clone the DNA or RNA sample provided and will detect for specific viral RNA sequences using color genetic markers and color sensors. The PCR has 3 steps in cloning DNA to prepare for detecting viruses.
First step is denaturation. Denaturing is heating the DNA sample to a higher temperature, around 95 degrees, to break apart the hydrogen bonds holding the double-stranded DNA together and separating it into 2 strands. The second step is annealing. Annealing involves cooling the separated DNA sample to a lower temperature of around 55 to 65 degrees Celsius that we can then add primers to. Primers are short synthetic DNA or RNA molecules of complementary strands that target a specific DNA sequence to serve as a starting point for the next step. The third and final step is extension. Extension involves heating up the sample again to a higher temperature, typically 72 degrees Celsius, to activate the Taq polymerase enzyme. During extension, the Taq enzyme extends the primer along the DNA to synthesize new complementary DNA strands to allow for the amplification of the viral RNA sequence that we are targeting, thus making it detectable in the sample.
To use this machine, you can start by taking a sample of the virus from your nose or throat using a cotton swab. You can then put the sample into a tube with some reagents. Then you can insert the tube into the PCR. This will amplify the viral RNA to make it more visible and easier to analyze during detection. Now we wait for around 1 hour and the result will inform you whether the patient has been infected with the virus.
You might be thinking, how would this affect our daily lives? Well, this can replace RT tests for a much more efficient and accurate detection of viruses. You can also clone any DNA sample of your choosing for any use case of the DNA you may find. Currently, you can make this PCR at home under guardian supervision if needed, or maybe in the future it will be able to be mass-produced and sold to the public for them to test viruses much more efficiently and accurately. Our PCR design can also detect any virus with a known RNA sequence, as it uses a much more streamlined 3-step process that can identify the genetic markers of any virus, whether it is discovered now or in the future. This is the end of our presentation. Thank you for your attention.